All right, chapter 3. In chapter 2, we discussed the people that contributed to Darwin's seminal work. But there were some things that Darwin didn't know. And aside from genetics, he did not know how old the earth was or how old things were. I mean, he had ideas, he took some guesses, but there wasn't the fossil record that we have today. So, people had heard of fossils by then, but it was relatively undeveloped. So, this chapter kind of expands a little more on that. How can we tell the age of certain layers of rock? What are some of the tools that are used? So at Darwin's time, most of the public again thought that they considered the Earth to be a few thousand years old. Maybe 4,000 years or 8,000 years old. Um, Geologists at that time thought it was much older, but of course, they're differing on how old it was. So some people thought many hundreds of thousands of years, or a few million years. They certainly didn't think of it in billions, right? So um, they had no idea really. Darwin, in his lifetime, he did try to take a look at it, and what he did was he tried to look at sediment deposits. When something's out, when something is just out, it gets covered with dust, and it gets covered with, eventually it'll get covered with dirt. And he looked at that in real time, like over years. Like he would keep going back to a site, and he would see how much it was covered by something. And then he tried to extrapolate that, and he tried to guess, um, he tried to make an assumption about how old Earth was. He was actually kind of accurate. He was thinking that the Earth was probably billions of years old, and there were people that disagreed with him at the time. And he had no proof. So this was a guy that really spent a lot of time um, trying to document everything and have like the scientific proof before he said something, right? So these were just things that he was thinking to himself, and he didn't really talk about it a lot. But anyway, there's one called Kelvin, Lord Kelvin, and he was one of the people that and he was a famous geologist at the time, and he disagreed with Darwin. And but his argument was, not that you date things based on fossils, but you date things based on heat. So there's like a whole temperature range that's based on Kelvin. Um, so anyway, that was his thing. He was more of a physicist, I guess you could say. So he looked at like the math of it. He figured, well, rock is going to cool at a certain rate. So if you heat it up, it's going to cool at like a fixed rate. So he assumed, and this was an assumption at the time, he assumed that the Earth, you know, uh, collided with the sun or was formed from the sun or whatever it was, that it was molten rock, and that as the Earth cooled, that was like a fixed rate. And so you could figure out how old the Earth was by just getting an accurate temperature of the surface of the Earth, and then just applying the math to it. And so he took his readings in mine shafts, because you couldn't do it at the surface, because during the day the Earth gets a little bit warmer, and then at night it gets a little bit colder, and it changes with seasons and stuff like that. So in deep down in the mine shaft, the temperature is going to be stable. So he took a bunch of readings there, and he came up with the idea that the Earth was around 20 million years old. But he was wrong. Why was he wrong was he didn't, I mean, what really wasn't known at that time was that, the center of the earth is not like a solid mass. Stuff's hot down there, and that stuff is coming up to the surface, it's going back down, it's constantly moving. So you're not going to get accurate readings. And in fact, you're going to get hotter readings than he would have thought. So he based all of his readings on the earth being kind of like a solid fixed sphere, when really the inside of the earth is fluid. I mean, it's literally fluid, but meaning that it's always changing. It's not static. It doesn't stay still. So because of that, he had faulty calculations. It was actually, his readings were not accurate, and so the 20 million years wasn't accurate. So let's go back to fossils. So fossils do have limitations. They're good. Like when you look at fossils, when you look at a lot of research, like the whole body of research, then you could start to have some confidence in your findings, right? But fossils in themselves, they do have some limitations. And so I listed here, some things that fossils have to go through, the vast, and by saying most fossils don't make it, I mean like the vast majority. You're thinking of, we have to think about things that, the animals were eaten and the microbes removed everything else and it's just bone. But that's not enough. You have to have something cover it. If something's not going to cover it, that bone is eventually going to decay and break down. And that's what happens to the vast majority of things that die. They do not become fossils. Water percolates through the sediment and it brings different minerals with it as it's going through, and it deposits it where the bone is, and it kind of works its way into the pores of the bone. And so as the bone kind of starts to decay, the minerals take its place. And so, really ideally, you don't want oxygen being exposed to the area, and it has to be that way for thousands of years. So if you have a good wind that comes along and blows the soil off the top, or some kind of water that comes and washes the soil off the top, anything like that, and that's it, it's ruined. It's not going to be a fossil. And Darwin himself understood that, fossils are imperfect, and fossils are shells and bones. So, what about soft-bodied? Now, there are some exceptions, but in general, what about soft-bodied organisms? Well, we don't have a good fossil record of those. There are some, but in general, it's not, it's mostly things with hard bodies. So there are some limitations, just because we find one thing in the fossil record, there might be lots of layers. There might be thousands or millions of years where, for whatever reason, fossils were never able to be formed. We have no way of knowing that, really. But anyway, going back to fossils, how can we tell how old these fossils are? Well, we use what are called isotopes. So this right here is an example of an atom. It's this one happens to be lithium. Not that you need to know that, you don't. But the question is, how do I know it's lithium? Well, in every atom has three parts to it. They have protons. So there's like this. So imagine like this planet, right? And then there's moons orbiting around the planet. That's kind of how I picture it. The moons are called electrons. And for this conversation, it's not that important. In the planet, you have protons and neutrons, right? So the neutrons here are the green dots and the protons are the red ones, and they fill the middle. The protons are what determines what kind of atom this is. And that's how I know it's lithium. I counted the red dots. There's three of them. It's lithium. It's always going to be like that. Lithium is always going to have 3 protons. It could have more electrons or fewer electrons. It could have more neutrons or fewer electrons. That's irrelevant. It has to have 3 protons only to be lithium. Now, usually the amount of neutrons in the middle is exactly the same. So if there's 3 protons, I'm going to guess that there's 3 neutrons, or whatever it is. If there's protons, if there's 8 protons, I'm going to guess 8 neutrons. There could be more or less, but usually it's like an equal amount. In this case, looking at this one here, we're counting 3 protons, and the neutrons are more. There's 4 neutrons. That's an isotope. So usually you have more neutrons than protons. And, what does it mean? Chemists, they could tell you. In biology, we don't really care so much. But at some point, it gets crowded, right? There's supposed to be 3 protons and 3 neutrons. That's kind of like the carrying capacity, right? And so now you have 3 protons and you put in 4 neutrons. Well, it's a little crowded. What if I put 5 in there? Now it's going to get really crowded, right? At some point, it becomes radioactive. At some point, you put in too much stuff in that middle, in that planet. It's called a nucleus. So when we cram too much stuff in that nucleus, it's going to break open. Now, let's say it breaks open and the neutrons flow out. Big deal. It doesn't matter. It's still lithium. However, if I lose any of these protons, it's no longer lithium. In this case, it'll be helium. So what happens when you lose the protons, when this nucleus gets too crowded, and why does it get crowded? Good question. I don't know. I know that if I look at all the lithium atoms in the world, most of them are going to have 3 protons and 3 neutrons. Some of them are going to have 4 neutrons. Some of them are going to have 5. Like, I don't know how many, but that's just the example that I got here. Lithium, every atom's different, but at some point it gets too crowded. The carrying capacity, what's too crowded depends on the atom, and we don't really need to know that, but it gets too crowded. And when this thing breaks open, and if one of these protons leaks out, that changes the atom, and now it's helium. So, so what we can do is that when you look at all the isotopes of any atom, there's isotopes of oxygen, there's isotopes of potassium, calcium, whatever it is, when you look at like all the carbon or all the oxygen in the world, some of it is going to be an isotope. Some of these isotopes are going to break open and they're going to leak the protons out, and they're going to become something else. For example, oxygen, when the protons leak out, it becomes nitrogen. So it changes whatever it is. When it breaks open and leaks out, it's a different element. It's a different type of atom, right? And so we know how long it'll take for half of the ion. We know how long it's going to take for half of the isotopes to break open. Let me try to explain it a different way. I know that, so let's take carbon, because we're all made from carbon. 99 or 98 point, let's say 99% of all the carbon that's in the world, think of like carbon dioxide, think of like all your food that you eat, that's all carbon based. You're carbon based. 99% of all the carbon atoms are regular carbon atoms. They're not isotopes, meaning that they have the same number of protons and neutrons. 1% are isotopes. How long does it take if I had a bunch of isotopes, how long would it take before half of them turn into nitrogen? And the answer is like right here, 5,730 years. So in 5,730 years, half of that carbon half those carbon isotopes are going to turn into nitrogen. So I can look at like a, a fossil of some kind of dead animal, and we expect there to be a certain amount of isotopes. Let's say 1%, right? So I expect 1% of those bones to be carbon isotopes. What if it's not 1%? What if it's only 0.5%? And I find some nitrogen in there now. Then I could say that it's 5,000 years old, 5,730 years old, because half of the carbon isotopes have decayed, and they've decayed into nitrogen. So that works a little bit, right? It works for a little while. You could measure something in thousands of years, but look at potassium. If you see how there's an expected amount of potassium that we expect to find in something, in a fossil. But if we find that there's less potassium and there's in fact more argon, we could look at the half-life, right? So I know this is kind of getting ridiculous here, but in 1 billion years, the amount of potassium is going to drop by 50%. So that's what the half-life means, right? So they just look at the proportion, right? We expect this much potassium. We found less. How much less did we find? How much more argon did we find than we expected? And you can figure out the half-life. And sometimes it gets a little bit crazy here, right? It's not, look, this is the half-life of this particular element. We don't think the Earth is older than 4.5 billion years, right? So, but we're just saying that you look at even like a slight decrease, you can approximate it. So this is radiometric dating. It's fairly accurate. And the other key is here, we never look at one fossil and make a judgment. You can't look at one fossil and say, oh, that fossil was 80,000 years old. Okay, maybe it was. That's one. But what if somebody else finds a similar fossil and comes up to a similar conclusion, right? Then the evidence starts to stack on itself. You start to have more evidence and you start to believe it more. So that's one thing. You can use radiometric dating. You can use biomarkers. So in rocks, we can look at certain molecules, and we know that some molecules are produced by particular living things. And I gave an example here of oganine. Oganine is a pigment, a color. It's a molecule. It's like a color pigment, and it's only made by this particular type of bacteria. As exciting as that sounds to you, it's only made by this bacteria. So we don't find oganine other places, just from this bacteria, right? So it's a sulfur bacteria, and as the name kind of implies, it's a bacteria and it likes sulfur. It loves to live in a sulfur environment. It doesn't breathe oxygen like other bacteria. So it likes to live in a place where there's like no oxygen and there's lots of sulfur. So you can look at it that way, like this bacteria like breathes sulfur, right? And it's rare to find these bacteria, but we do find them. They are around. So if you find a rock, for example, they found this pigment inside rock. So we have this pigment, oganine. It was found in this rock that was dated at about 1.6 billion years old. So we don't ever find this pigment anywhere else except for in this bacteria. So we assume that in this rock was this bacteria. And since we know that this bacteria does not like oxygen and does like sulfur, that suggests that maybe the atmosphere back then was high in sulfur and virtually no oxygen, or very low in oxygen. And it really that's an example of using a biomarker to give more strength to the notion that the Earth wasn't always filled with oxygen, and it wasn't, by the way. So when was the earliest life? Um, how can we have an idea about it? Well, isotopes, or I'm sorry, fossils, they can go back, but you need something a little bit more than that. And so in these crystals called zircons, they're pretty good at storing isotopes. And so carbon carbon isotopes can come from things that were living, but they can also come from things that never lived. So like take carbon dioxide, right? That's not living and it never lived. So that's something different, right? So but you can tell the difference between biotic sources and abiotic sources. The carbon the carbon isotopes that come from living things are different from the ones that come from things that never lived. Right? So things that lived or used to live, they prefer isotopes with like fewer neutrons, right? So like a carbon has 6 protons and 6 neutrons. So biological molecules will have carbon with like 6 protons and 7 neutrons, or maybe 8 neutrons, but not more than that. Carbon isotopes that come from like non-living things, they'll have like 9 or 10, right? So you could tell the difference. You could tell which one is which. So back in around 2004, they took carbon from like these really old rocks, 3.7 billion years old. That's what they were aged at. And we found carbon isotopes in them. That's not the part that's interesting. The fact that is interesting was that they were the lighter carbon isotopes that we associate with living things. So that suggested that things were living all the way back 3.7 billion years ago. So some people, after those findings came out, some people agreed, some people disagreed. They thought, you're measuring something, you're measuring carbon that's like super old. It might have been from some other type of geological process. It might in fact be abiotic carbon, and you're just not reading it right. But anyway, one researcher, Atwood, working in Australia, found something called stromatolites, which are bacteria, and she found this bacteria in rocks dated around 3.5 billion years ago. We have strolomites today. So if you look at the other PowerPoint, you'll see some photos of them. So we have it today. It's not like super common, but there is strolomites today. And they found strolomites fossilized in rock. And you can see that it's the same stuff, right? So then the question is, how old is the rock? Well, the rock was about 3.45 billion years ago. Bacteria is a living thing. So she deduced that this is the first living thing. And there's a lot of people that agree with her. And it's most likely the strongest evidence that, it's the strongest evidence of life existing that long ago. Darwin, on the other hand, in just a couple hundred years ago, he had no idea. There was no fossils really confirming evolution or the age of the earth, or they didn't have the technology. They certainly didn't have technology like biomarkers. I mean, no, absolutely not. That didn't exist. Radiometric dating, that wasn't around until the 1900s. We didn't even know about atoms until, until 100 years ago, or maybe 110 years ago. So we didn't even know about atoms then. So we couldn't do any of these things with fossils back when Darwin was around. So now, since the last, let's say 40, 50 years, honestly, since the 1900s, because it's one thing to know about DNA and have the technology, but it's another thing for the technology to be cheap enough for us to mess around with. And now it finally is, right? Starting in the 90s, we've been able to take DNA sequencing and like look at things, right? And create these family trees, try to figure out what came from what and how old things are, right? We can look at fossils, and that's one way to figure out how old things are. We can also, and you can look at the layers of fossils, right? So things that are buried deeper, you would deduce are older than things you find closer to the surface. But another thing we can do now, especially since the 90s, is that we can look at the DNA. And so based on that, we've made 3 groups, and this is all still kind of new. We have 3 groups when we look at the family tree. And these big 3 groups are archaea, bacteria, and something called eukaryotes. So archaea and bacteria are very similar. Archaea are actually rather new. When I was in school, I didn't even study archaea. I didn't know about them. So that's how new it is. But anyway, they're kind of similar to each other. They're both single-celled organisms, meaning they're made out of one cell, done, right? So, but there's some differences between them, and honestly, you don't have to know the differences. I just wanted to show you that they're different, but just kind of know that they're similar, but they're slightly different. But they're both really small. They're about 1/100th the size of our cell. So let's look at it a different way. There's bacteria. There's like their close cousins, archaea. And then there's everything else that's living. Everything else. So think of like a little amoeba swimming in a pond. Think of a fish. Think of a monkey. Think of a plant. They're all eukaryotes. Everything else is a eukaryote but bacteria and archaea. So eukaryotes can be single cell, and I gave you an example of, like I said, an amoeba, paramecium, something like that. But most of them are multicelled, like us. We have trillion cells. By the way, to tell you something about bacteria, there are more bacteria in your body than you have cells. So you have about 1 trillion cells in your body, or that's what makes up your body. Bacteria outnumber you. So bacteria are found absolutely everywhere, right? They're found in the most inhospitable places that you can imagine, and they're found inside our body. It's kind of a new branch of science because what do these bacteria, or what do like the microbes in us, what do they do with us? Are they flipping enzymes on? Are they causing us to change? Are they causing us to maybe start to, are they turning genes on to get a particular disease or not get one? This is all kind of new. We have 400 species of bacteria just in species, 400, not 400 bacteria, 400 types of bacteria in our gut, just in our digestive tract. This is all kind of a new area of research that we really don't know about them. They're living symbiotically with us, right? They're doing things for us. We're doing things for them. Sometimes they make vitamins for us, and we give them energy. So anyway, let's get back to, so bacteria and archaea it's pretty much that they're the oldest living things. And then from there came eukaryotes. And eukaryotes are a little bit different in that our cells have a nucleus. That means that our DNA has a membrane around it. So let's look at the age of some things. Bacteria are considered to be the oldest of everything living. They were the first living things to come around 3.5 billion years ago. So if we are to say that the Earth is around 4.6 billion years old, so you have about 1 billion years where there was nothing living, and then bacteria came. We don't find the first eukaryotes, that's our type of cells. We don't find those until 1.8 billion years ago. But still, when you look at the earth today, we are living in their world. Microbes, although we don't realize it, microbes dominate everything. If you were to take the weight of microbes and the weight of everything else, like all the other living things, microbes beat us. So microbes still dominate. They have the most weight, the most number, the most diverse. They are everywhere. It's really their world, and we just don't understand it and we don't realize it. But anyway, moving on. The first type of multi-celled eukaryotes, multi-celled things that were not bacteria, came around 1.6 billion years ago. And then you see here I have some kind of multicellular algae because nobody knows what it was. And I know this is sounding super boring at this point, but I'm just kind of showing you, how old these things are. And let's move on a little bit more. When did animals start to come around? Well, sponges are the simplest animals, and you'd be thinking sponges are totally not animals. And I could see how people would think that. I would think that too. But we do share similarities with them. The way they process food, there are similarities, and there's genetic similarities that sponges have with us that are not found with other animals. So the first animals, therefore, around 650 million years ago. So now we're in the millions. Larger organisms that can move around, because obviously sponges are fixed. Larger organisms that can move around about 575 million years ago. When did we start to develop as larger organisms? When did we start to get backbones, nervous systems? About 515 million years ago. That's when chordates came around. So that's what we are. That's like our phylum. We're called chordates because we all have backbones. Dinosaurs were around 230 million years ago, and their extinction happened around 65 million years ago. Not that I want you to memorize all these numbers. No, you don't have to. I'm just kind of showing you a rough idea of when things started to come around. And I want you to see that some of these things are not actually, when you look at the age of the planet, 4.5, 4.6 billion years, and you talk about dinosaurs 230 million years ago, that's relatively recent. Angiosperms, plants with flowers, 130 million years ago. Plants without flowers are a little bit older than that. Mammals didn't come on the stage until like 60, 70 million years ago. And then when you got a diversity of mammals, and think back to chapter 1 when we talked about the whales and dolphins, we're talking about 50 million years ago. Hominids apes, monkeys, things like that, around 7 million years ago. And then finally, the humans, we estimate somewhere around 200,000 years ago, which seems like a long time until you look at it, when you look at the whole age of the Earth. So if we were to take the age of the Earth as a clock in one 24-hour day, humans came around about 5 seconds to midnight. So really not a long time when you look at, everything, when you look at the age of the Earth. Anyway, so some of the stuff in the last slides, we're going to talk about in a little more detail. So we're going to, in the subsequent chapters, we'll discuss things like bacteria. I will try to make it as easy as I can. So, but the nature of it, some of you have already fallen asleep. So it's some of it's difficult. I'll try to put a lot of photos in. Okay, so we're going to talk about bacteria. We're going to talk about different eukaryotes. We're going to spend a couple chapters on that. So that's coming in the future. And I will end the meeting here.